Now that we've been through chapter 10 and talked more about the chemical nature of DNA, we're going to now move into talking about how to copy that DNA and synthesize an exact replicate of that DNA. So, chapter 12 is all about that replication and recombination process. Some of the basic rules of replication are that DNA replication is actually conducted in a semi-conservative manner, and which means that you are not going to have if you're synthesizing one strand of DNA, then the actual copy is not going to be verbatim what was in that original strand of DNA. It's going to be the complement. So it's a semi-conservative rather than a fully conservative type of replication. The second basic rule is that replication is going to begin at origins of sequence. So those origins are going to be different depending on the type of replication that takes place, and we'll talk about that here in just a second. The third basic rule is that this process of DNA synthesis is going to be initiated by very short segments of RNA. So again, DNA synthesis is going to be initiated by short segments of RNA, and those RNA components are called primers. The fourth basic rule is that as a DNA strand elongates, and we add more nucleotides to the 3' end, it's important to remember that DNA, the addition of nucleotides is always going to happen on that 3' end, and that's because where the available OH group is at. So this is why we spent so much time in chapter 10 talking about the orientation of the DNA strands, their 5' versus their 3' direction. Basic rule number 5 is that the replication process is continuous in the leading strand, but discontinuous in the lagging strand. Don't worry about that too much right now. We'll explain it again in a minute. And then the last rule is that the new strands of DNA are going to be complementary to the old strand of DNA. There's 3 basic types of replication that can take place depending on what type of organism we're dealing with. If we're dealing with bacteria and protozoa that have circular DNA, the type of replication that takes place there is called theta replication. There is, depending on the bacterial organism, there's also another type of replication that can take place with a circular genome, and that is circle or rolling circle replication. And then the type of replication that we're going to spend a lot of time talking about that we're most familiar with is linear eukaryotic replication. And we'll look at examples of those in just a few slides. So there's also 3 major requirements of replication. The first one is that the template actually has to be single-stranded. So we know that DNA is stable in a double-stranded fashion. However, for synthesis to take place, there's a process that it has to go through to separate from itself so that the synthesis can actually take place. Second major requirement is that there has to be a bunch of substrates ready to go to assemble to help with this process. And thirdly, those substrates, some of those may be in the form of enzymes and other proteins those are going to help read the template and then assemble that template into a newly synthesized substrate of DNA. So one of the biggest concepts that you guys need to have a good handle on is that DNA synthesis always occurs in the 5' to 3' fashion. 5' to 3', 5' to 3'. Just start getting that in your head so that as you are thinking about different types of replication and you're working through different problems and trying to match up nucleotides, that you can kind of get a handle on DNA is always going to be synthesized in the 5' to 3' fashion. So in this figure here, we've got our template strands. We have 2 template strands. They're shown in gray. Our template strand on the top here has a 5' at one end and a 3' at the other. The complementary strand has the 5' at the across from the complementary strands 3' and ends with a 3' end down here. So again, those are the gray DNA strands in this figure. The red strands in this figure are the newly synthesized DNA. And just keep in mind that on this top template, we are going to be adding nucleotides together in a 5' to 3' direction. And then on the lagging strand, that'll be reversed. So we'll start over here to the left with the 5' and adding on nucleotides in a 3' fashion. So here's some figures of the different types of replication that can take place. Again, that theta model has to do with bacterial systems with circular chromosomes. And the leading strand is going to be complementary to the original template. And it is going to be synthesized in a 5' to 3' direction. Some bacterial species will actually replicate with a rolling circle model, and this is where that double-stranded DNA is going to be stabilized in this circular model. However, at the point of DNA origin, we're going to start synthesizing a new strand of DNA. And that whole process is just going to keep going until it gets to its ending base pairs, and then you'll have a new chunk of DNA that will then be able to separate out through cytokinesis and cell replication. And finally, the last type of replication that we're going to spend a lot of time studying is linear eukaryotic replication. And this is where the chromosome material is going to be in a linear fashion rather than a circular fashion. And those DNA strands, template strands, are going to be oriented in the 5' to 3' position. However, we're going to add new nucleotides in that new sequence from a 5' to 3' direction. Okay, so this is one important note to take into consideration here is this leading versus the lagging strands. So our leading strand is going to be the strand that has that is going to add nucleotides to that 3' end, and then the lagging strands are going to be taking place behind that leading strand. And these lagging strands are usually replicated in smaller chunks, and so you get a chunk of DNA, a gap, another chunk of DNA, a gap, another chunk of DNA. And what these different chunks represent are called Ozaki fragments. And those Ozaki fragments, they are truly DNA material, but they're going to, at the end of the replication process, there's going to be some binding that takes place between those to link them back up. So let's talk more specifically about the mechanization of replication. The first step is that we've got some initiator proteins that are going to bind to the origin of replication and going to cause DNA to start unwinding. And helicase is one of those enzymes, and DNA helicase is going to bind to the template DNA strands, and then it's going to help unwind the DNA. And we're going to, as soon as the DNA starts to unwind, these stabilization proteins that you see in purple here are going to come in and bind to each individual template strand to help stabilize the DNA. Because remember, DNA is still negatively charged because of the phosphate group that's a part of the nucleotide, so it needs to be stabilized, or the strands are going to just go back right into the double-stranded helix, and synthesis won't be able to take place. A few of the enzymes that are involved in this replication process is DNA gyrase, which is a topoisomerase, which is going to bind a little further downstream in the double helix form and is going to help absorb some of the effects of that supercoiling that's going to take place from uncoiling the two template strands of DNA. And then the primers that are actually going to help out with the replication process. The primers are all DNA polymerases, and there's a whole family of them. We'll talk about them again in a minute. And they each are going to require a nucleotide that has an OH group on the 3' position. And finally, one of the last enzymes that are involved is DNA primase. And primase is made up of RNA, not DNA. And primase is going to synthesize short stretches of RNA that is going to make that 3' OH group. So the polymerases that we talked about in the previous slide, there's a whole family of them, but in general, DNA polymerase is going to attach to the available OH group on the 3' position and go through a polymerization process. And that process is where we're going to start seeing addition of nucleotides being brought in on that 3' end. So this figure shows the whole process that we've previously looked at in small chunks. So this is a really good place to start to begin to draw this out, try to get a handle on what enzymes are coming in at what time. And you guys are going to spend some time on this in activity as well. So I want to talk just a little more in depth about this polymerase family. In linear eukaryotic systems, DNA polymerase 3 is actually going to be the enzyme that is going to add nucleotides to the new strand. Whereas DNA polymerase 1 has a different function. It's going to be the enzyme that is going to remove RNA primers and replace it with the DNA. So keep in mind, RNA, the primers are always going to be RNA. They're going to be they're going to set in these new strands as kind of a buffer transitionary period, and eventually DNA polymerase is going to come in, pull those out, and replace them with DNA so that you have a full DNA strand. During that polymerization process, there's going to be a nick that ends up happening, so a gap that exposes the sugar phosphate backbone, and another enzyme is going to come in and actually go through the sealing process and create a phosphodiester linkage there. And so that's DNA ligase is the enzyme that's responsible for that. So again, here's our basic rules of replication. Make sure that you have a really good handle on these because we're going to talk about each one of them more in depth in the full lecture.